Yeah, hi, hello, welcome to our YouTube channel. Myself, Sendil Kumar, biology teacher. Okay, today we are going to deal a very important chapter, biotechnology, principles and processes in grade 12, CBSE. Okay, so what is biotechnology and what are the techniques which is involved in the biotechnology and how we need to follow the basic principles and the processes in the technique. That is what the thing we are going to deal in this particular chapter. Okay, so what is biotechnology? It is the technique of using living organisms or their enzymes for product and the processes useful to human being. Okay, so for this we'll take a very good example called fermentation. What is fermentation? So we'll deal with the example. If you take grapes, grapes used to be converted into wine. This process is called fermentation, but this conversion, the fermentation can be achieved by living organism called yeast. So here yeast playing a very important role. The yeast plays a very important role of converting this particular substrate into product called wine. Okay, so the yeast in the sense here it releases some enzymes also. Okay, so here yeast as well as its enzymes act as a catalyzer. Okay, which is act as catalyzer and act on a substrate. Who is a substrate? Grape. The substrate is actors, grapes that will catalyzed by a yeast and finally you will get a product called wine. Okay, so this is a one kind of technique. If we go with another example, for example, curd. So the curd, how we get curd? The milk will be converted into curd. That process is called curdling with the help of lactobacillus. So here the lactobacillus is a kind of microorganism which act on a milk and converted into curd. That process is called curdling. So here the microorganism and its enzymes, okay, that play important role to convert the substrate into product, and also the overall the function is called processing. Okay, the processing in the sense the downstream process is that it is kind of sterilization techniques and all that one by one we are going to deal in this particular chapter. Okay, and see, so about the biotechnology, the European Federation of Biotechnology, this is one association who has given a definition about biotechnology. So what is that means? The integration of natural science. The integration of natural science, organisms, cells, part of thereof. Okay, in the cells part in the sense, so this is animal cell, nucleus, endoplasmic reticulum, okay, centrioles here you could identify, and Golgi apparatus and some ribosomes. So all those things, mitochondria, these are all the cell organelles, right? So that is what here represent as parts of thereof and molecular analogues. What is molecular analogues? It's nothing but genetic material. Is nothing but genetic material. The molecular analog is nothing but genetic materials. Okay, for these are all we are using in biotechnology for products and services. What are all those products and services? We are going to deal one by one. Okay, and see, so especially the biotechnology, already I said this is a branch of biotech, branch of okay, the biotechnology is the branch of science. Under the umbrella of biotechnology, the various kind of techniques we are going to deal. So some example we are going to discuss here. What are that? The microbes mediated processes. Okay, for example, making curd already we have discussed, and bread, wine. Okay, so these are all can be achieved. This is all the products, right? Curds, bread and wine and all product. These products and all we are getting through microorganism. What are the techniques we are following to get such a product? The techniques comes under the umbrella of biotechnology. Okay, and the in vitro fertilization, this is a test tube baby program. So why they go for in vitro fertilization means if the male or a females are impotent, okay, impotent in the sense in a male azoospermia, in especially in a semen the sperm count is less, or in a female the egg developmental problem or any kind of issues take places. In that sense they have to go for a in vitro fertilization. Okay, not detailing we are going to discuss about the what is the reason behind in vitro fertilization and all. So what is what, because this technique comes under biotechnology. Okay, so the in vitro in the sense it is nothing but under laboratory condition. Okay, in a laboratory condition, in a aseptic environment we are doing a test tube what is it, test tube baby programs. Okay, and next, synthesis and using of a gene for expression of the particular protein. Okay, for expression of particular protein, here we are using the various kind of techniques. Okay, so various kind of techniques like a gene therapy to express the particular gene. Okay, so that is what using of gene to synthesize the particular enzymes or protein, that also we are going to deal. And preparation of DNA vaccine. Okay, so when we talk about DNA vaccine, nowadays we are very prone to the corona, we are facing a lot of the problem because of that, and especially the coronavirus, just a rough diagram I am making. It has a structure like this. Okay, inside you could identify the genetic material. It has a RNA. RNA is the genetic material of coronavirus. So here the entire structure, the entire physical structure is not responsible for causing disease to human. Okay, in that maybe the particular part or maybe the particular protein may cause, and it is responsible, sorry, the particular protein is responsible for a disease, that is act as a foreign substance to our body. Because of that we are getting some diseases, some problems. Okay, so here what the scientists did, so whatever the part of a microorganism or a pathogen which is causing a disease to a human being, that particular gene which is responsible for causing a disease, that means here the protein, for example protein, one particular protein is responsible for causing disease, means this can be expressed by a particular DNA sequence or it is considered as gene. Okay, the particular gene is responsible for expression of particular protein. Okay, so the what scientists did means they are isolated the particular gene and it can be utilized for a DNA vaccine. Okay, the particular gene has been utilized for a DNA vaccine. Okay, and next, and the correcting the defective gene. That defective gene can be replaced by a various kind of technique is one of that thing is gene therapy. The gene therapy is a therapeutic treatment we are using to treat any genetical disorders. Okay, it may be a somatic cell. Somatic cell gene therapy is there, and germline gene therapy. Just I'll put JT, gene therapy. Okay, like that the two types of gene therapy is there, that now we are going to discuss in the upcoming videos. Okay, so let us move to next content. So here the core technique of modern biotechnology. In the modern biotechnology, What is considered as a core technique means here the genetic engineering, and second one is a bioprocess engineering. Okay, so what is genetic engineering? Actually, it is a technique in which the genetic material like a DNA and RNA is chemically altered and introduced into host of the organism to change the phenotypic character. Whatever the phenotypic character we want to change, that we can able to do with the help of the technique called genetic engineering. For example, we can take BT cotton. Okay, so this is a genetically modified plant. Okay, or transgenic plant used to say. Okay, that can be done by a genetic engineering. Okay, in detail we are going to deal in the upcoming videos. Okay. So here next, bioprocess engineering. What is bioprocess engineering in the sense? So the maintenance of the sterile ambience in a chemical engineering processes for growing a desired microbe, that is ultimate aim. Previously we were discussed, right, from what is making a wine. Okay, so the grapes will be converted into wine. There what microorganism we are utilizing? Yeast. Okay, so for the fermentation process, especially for the fermentation of the wine, the process name is called fermentation. From that we are getting product called wine. Okay, in that sense we are in the need of microorganism in a large amount. Okay, the wine preparation and all used to be in a large amount. Okay, so the desired microorganism supposed to be cultured and eukaryotic cell in a large quantity for a manufacturing of antibiotics, vaccines and enzymes. So these are all the desired microorganism. The specific specific eukaryotic cell. Okay, that means microorganism for antibiotic, vaccine and enzymes. We need a pure form of microorganism. For that we need to maintain a sterile environment. Okay, without maintaining a sterile environment, we cannot able to get a pure form of microorganism for a desired techniques. Okay, so whatever the desired techniques, whatever we are planning to do any particular product. Okay, for example, wine or a bread. Okay, or maybe a curd. For that and all we need a specific microorganism. If along with that any other microorganism has been added, then the particular product may contaminate and we may not get a desired quality of the product. Okay, so to avoid that they go for a bioprocessing engineering technique called, mainly it is nothing but a sterilization. Okay, to sterile ambience, to keep the sterile ambience, this particular bioprocess engineering was there. Okay, and next here some basic steps in genetically modifying an organism. What are the basic steps to be followed to do the genetically modifying an organism? Okay, so what are the steps means? Identification of DNA with desirable gene, whatever desirable gene. Okay, whatever our gene of interest, that is called desirable gene. Next, introduction of identified DNA into the host. Introduction of identified DNA into the host. Whatever our desired gene we want to introduce into the host. For that we are going to use vector. About that we are going to deal. Wait. And next, maintenance of introduced DNA in the host or transfer of DNA to its progeny. Whether whatever our desired gene has been inserted into the host and it is transferring to the next progeny, that means next generation. That also we have to take into the consideration. These are all the things to be followed in the for a genetically modifying an organism. Okay. Next, see. First, we were discussed, right? Identification of DNA with the desirable gene. So here, especially in a old traditional hybridization method, the techniques leads to the inclusion and multiplication of both the desirable and the undesirable and the desirable gene. Okay, both the desirable and the undesirable gene. Okay, which is which is not required, okay, which is not required, that particular gene along with our desired gene has been included or inclusion, okay, it has been included in a hybridization technique in old traditional method. But in a genetic engineering, only the desirable gene are introduced, okay. So here now we are identified our desired gene, okay, we got a desired gene. Next, the introduction of the identified DNA into the host, how we can introduce, how we can transfer. We also need of particular vector to transport our gene of interest. So here such a plasmid, the vector DNA such as the plasmid is used to deliver an alien piece, the alien piece of DNA. The alien piece of DNA is nothing but our desired gene or our gene of interest. All are same. இது மூணுமே ஒண்ணுதான், சரிங்களா? alien piece of DNA na for vector, especially vector DNA யாருக்கு? plasmid. The plasmid vector where you could identify, especially in a bacteria. In a bacteria, this is a plasmid DNA. This is bacterial DNA or this is called as main DNA. It is also called as main DNA ன்னு சொல்லுவாங்க, சரிங்களா? So along with that, the plasmid DNA also is there, okay. In the plasmid DNA, especially in that plasmid DNA, we are using as a vector to transport our gene of interest. For that particular plasmid, it is a alien piece, okay. For a plasmid, it is a alien, but for us, it is our gene of interest, our desired gene, which we are going to, what is it, introduced into the plasmid, okay. So that is what. Then after it has been inserted into the plasmid vector, then it has to be transferred to the host organism, okay. What host organism? One by one we will see. And see, now we have been inserted our gene of interest into particular plasmid, okay. Here just consider this region, this is our gene of interest. Our desired DNA, our desired DNA, gene of interest, okay. For a plasmid vector, it is going to be a alien or a foreign DNA, okay. And now a piece of alien DNA has no the sequence called origin of replication. That is a drawback, okay. Whatever our desired gene we have taken, for example, this is what the fragment of DNA, this we have taken, okay. Why we have taken means, for example, this is for a insulin, okay. This gene is responsible for expression of insulin, okay, to make a humulin, okay, to make humulin is what? Insulin, okay. It is nothing but a insulin. To make that particular insulin, we have taken a particular gene of interest, okay. So that may not have a origin of replication site, okay. So if don't have a origin of replication, cannot able to replicate. In that sense, what we have to do, we have to introduce into the vector which has a origin of replication, okay. So what we did, so it is cannot multiply itself in the progeny cell of the organism, okay. So I will repeat again. So here, so the alien piece, it don't have a origin of replication site, so it cannot able to start a replication, so it cannot multiply itself in the progeny cell of the organism, okay. It cannot multiply inside the progeny organism and it cannot able to transfer to the another organism also. So for that purpose, what we have done, hence the alien DNA is integrated into recipient genome who is receiving, okay, recipient genome, it multiple recipient genome which has origin of replication, especially here who has the origin of replication means the particular plasmid vector, it has a ORI, just I mentioned, okay. Again I will mention here ORI. So the origin of replication region. Okay. So here the origin of replication region is exist in plasmid vector. So where gene of interest has been inserted, so then it can be transferred to next progeny. How it will be transferred to next progeny, sir? You might have a doubt. So let us deal with that. First, here if you take one particular sequence, okay, here we got a plasmid vector inside E. coli. This is a E. coli bacteria and this is a main DNA and this is plasmid DNA. And this is a genetic material which we have been isolated from a particular organism. Already we have discussed. So we are in the need of particular gene which is have a capability to express insulin. So what we supposed to do means, in lower class you would have studied, we supposed to use the restriction endonuclease RE. Restriction endonuclease. This is an enzyme which can act on both the genetic material which we have isolated, okay, as well as in a plasmid DNA. Okay, it will recognize the particular sequence and it will be fragmented. A restriction enzyme, it will be break into small fragment. Okay, so that's why it is called. It is also called as molecular scissor, used to say. Okay, it used to cut the DNA into small fragment. Once you got a fragment, here also we are using the same kind of restriction endonuclease, so that you may get a structure like this. Okay, here you could identify cap. And just consider, just I make a for understanding, I make a different color. Okay. Okay. So here this fragment we are taking and this particular plasmid vector, in between that we are inserting our gene of interest. We are inserting our gene of interest. Now it is called rDNA, recombinant DNA. This recombinant DNA, whatever we have discussed, right, about we are inserting into the plasmid vector. Okay, that is what in a simplified manner I am explaining for understanding whatever the content we are discussed here now. Okay, so here now it became a recombinant DNA. We are inserting back into the same kind of So here I will draw. So this particular recombinant DNA, we are entering into the host of the same host called E. coli. Okay, same host E. coli. Now there is a recombinant DNA. Okay, this is a rDNA. This is a rDNA which we have inserted into the particular host. Okay, this host is nothing but E. coli. Okay, after inserting, this particular host has been transferred into the culture medium, petri dish. Okay, transfer into the culture medium called a petri dish. Okay, inside the petri dish or even a conical flask, okay, we can culture or whatever the transform the host. Okay, so once it has been recombinant DNA has been transformed, they are called as transformant. Okay, this particular host is called as transformant. This particular E. coli, again it has been, what is that, inoculated or transferred into the culture medium for a further reproduction. Okay, so then what happens, it will take only 20 minutes. It will take only 20 minutes to producer to progeny. Okay, it get only the 20 minutes to produce progeny, offsprings. Okay, so that is what the beauty. Along with that particular the progeny, our gene of interest also replicate. Okay, so this is our ultimate aim for multiplying our gene of interest. Okay, and let us continue now. So here the process of joining, what is recombinant DNA? That is what we are going to do. What is recombinant DNA? The process of joining and inserting a foreign piece of DNA. This is what we already discussed, into a host organism to produce new genetic combination is called recombinant DNA technology. Okay, we are recombining. We are gene of interest, we are inserting into the plasmid vector with the help of restriction endonuclease. The restriction endonuclease is an enzyme which cleave the particular base pair, okay, of both the DNA as well as the DNA of plasmid. Okay, so after that we are inserting our gene of interest, that is called recombinant DNA. Okay, and the first recombinant, in that way, the first recombinant DNA was produced by Stanley Cohen and Herbert Boyer in the year of 1972. Okay, so what they did means they isolated an antibiotic resistant gene. What is the antibiotic resistant gene? So the bacteria which have a resistant to the particular antibiotic, like a tetracycline, ampicillin, kanamycin, okay, streptomycin, like that. The various kind of antibiotic is there. Some microorganism, they are resistant to that. Okay, in that way they have identified the one particular plasmid which is have a antibiotic resistant gene. Okay, that particular piece of DNA from that plasmid called plasmid of what? Salmonella typhimurium. Okay, this is a bacteria. From that bacteria has a main DNA as well as the plasmid DNA. Okay, so in that sense, here from the Salmonella typhimurium, the scientists have isolated the antibiotic resistant gene. Okay, and it was linked with the plasmid vector and transferred into the E. coli. How they link? As usual, we have discussed, right? Okay, this particular gene of interest, the antibiotic resistant gene, this particular gene, okay, the piece of DNA we are discussing, right? This is considered as gene of interest. That has been isolated and inserted into the plasmid vector. Okay, once it has been inserted, that will become a recombinant DNA. That recombinant DNA has been transferred into the E. coli. Okay, as a result, the gene was expressed and multiplied in E. coli. Okay, once it has been inserted, that whatever the genetic information which is exist in gene of interest, that will express. Okay, once it has been expressed, that can be isolated by a various kind of technique. Okay, that one by one we will discuss with the new videos. Okay, thank you so much. Bye bye.